Welcome everyone. Today we are going to learn about ATP driven pump. Now at first I want to mention that there are three major categories of transport protein is present. One is channels, second is transporter. Under the transporter there are uniporters, symporters and antiporters. And the third category is ATP driven pump. Now ATP driven pump contains four different class of proteins. One is called P class pump, V class pump, F class pump and ABC transporter. Now today we are going to learn about F class pump. At first I will mention some basic facts about this F class pump. The first point we have to remember that this F class pump is present in the three locations. One is bacterial plasma membrane, in the bacterial plasma membrane, then inner mitochondrial membrane, and in the thylakoid membrane of chloroplast. So in these three locations you can find the F class pump. One is bacterial plasma membrane, inner mitochondrial membrane and the thylakoid membrane of chloroplast. In this video I will explain the F class pump that is present in this inner mitochondrial membrane. But remember, the structure and function of the F class pump that is present in all these three locations actually very similar. So if you understand one F class pump structure and function, you can understand all the F class pump that is present in these three locations. So the first is location, where it is present. Second point you can remember, that is this F class pump only transport remember this, very important, only transport protons, that means H plus ions. Another thing is these protons, this is very important, these protons move down the remember this proton electrochemical gradient. Now if you have to understand this factor, proton electrochemical gradient, at first, because this information is so important for the functioning of F class pump. So to understand this, what is proton electrochemical gradient, I have to draw a membrane. It is not plasma membrane, it could be a plasma membrane, but suppose this is a membrane. And we know that all biomembranes are actually have two lipid bilayers. So suppose this is the two lipid bilayer of the membrane. And one side of the membrane is called the exterior side of the membrane, and one is called the interior. Okay, suppose this is interior. In case of inner mitochondrial membrane, this exterior side is the intermembrane space, and this side of the membrane is the matrix. I will draw the mitochondria, then you can easily understand. But for the understanding of this proton electrochemical gradient, just I am mentioning, suppose this is the lipid bilayer, the exterior is the intermembrane space and interior is the matrix of the mitochondria. So what happens here, in case of mitochondria, the intermembrane space contains more hydrogen ions outside, and inside is less hydrogen ions. Now this is called the concentration gradient. We call this difference in the protons concentration in different portions of the membrane, we call it chemical concentration gradient. Basically in my all transport videos I have mentioned there is two different movement occurs by the molecules or ions during this transport. One is called downhill movement, and another is called uphill movement. This is very important. And you can understand this downhill movement and uphill movement by actually this explanation, that suppose this is a hill, and if someone try to come down the hill, we call it downhill. Okay, and we know that during downhill movement, human also feel easy and it requires less effort. But during climbing up of a hill, we call it uphill, it requires more energy and more effort. The same is true for molecules or ions also. When molecules move from higher to lower concentration, we call it downhill movement. And during this movement, no energy is required. That is why it is called passive transport. Passive transport. Not only that, during this transport, energy is actually released. So this is called downhill movement. In case of uphill movement, molecules or ions move from lower to higher concentration. That requires energy, and that's why it is called active transport. Active transport. So here what is happening in case of this proton electrochemical gradient? At first I want to draw the F class pump. The F class pump has two domains. One is this domain I am writing here, that is the FO domain. This domain actually travels the membrane, so we call it membrane spanning domain. We call this membrane spanning domain. Why it is called FO? FO means oligomycin sensitive. This is not zero, this is FO. O means oligomycin sensitive factor. Okay, sensitive factor. And there is a globular structure present, or sphere-like structure present in the matrix site. This looks like this, and this is the F1 of the F class pump. So F class pump has actually multi-subunit proteins, but you can divide it into two subcomplexes, FO and F1. Now protons actually move. These protons actually move from higher to lower concentration. From here to it moves into inside of the matrix. And I have mentioned that when ions or molecules move from higher to lower concentration, this is called passive transport. So protons actually show passive transport, and it releases energy. This is the chemical concentration gradient. Okay. Now, not only this actually helps in the driving of protons inside the matrix, but there is another factor works, that is called the membrane potential. Remember this, another fact I want to mention here. That is membrane potential. Membrane potential, or you can remember it as electrical potential. Okay, that means there is a charge separation occur. As I have mentioned that in the exterior or intermembrane space of the mitochondria, there is more H plus ions present. So that means more H plus positive ions are actually present here. More positive ions are present here. And that's why there is less positive ions present. That means there is more negative ions present inside. And that is called the electric potential or membrane potential. In case of mitochondria, this value of this membrane potential, electric potential, is minus 150 millivolt. And the chemical concentration gradient actually denotes the concentration difference of the protons. In case of mitochondria, the intermembrane space has a pH value of 7.4 and inside is pH is 7.9. So there is a very small difference, but there is a difference in the pH concentration that contributes to the value approximate minus 30 millivolt. And togetherly, if you combine these force together, you will find 180 millivolt. When these two factors actually acts togetherly, we call this electrochemical gradient. Remember this, electrochemical gradient, because here chemical concentration gradient of the proton and the membrane potential or electric potential combining to move these protons inside our matrix. Now, as I mentioned that ions move from higher to lower constant, it feels very easy, so it will move. Not only that is the driving force, another driving force is the negative charge inside the matrix. That's why the positive protons are actually attracted towards the inside more. So these two factors combine to make the proton electrochemical gradient. Another thing is we can also call this proton electrochemical gradient also by proton motive force. Proton motive force, in short we call PMF. This is very important. So protons actually move from intermembrane space to the matrix, and by this way it actually generates ATP. From ADP and Pi, it makes this F1 portion makes the ATP. Now this mechanism, that movement of protons actually helps in the generation of ATP, is called chemiosmotic coupling. So we have to understand this point also. So there is a chemiosmotic coupling occurs. Chemiosmotic coupling occurs. Now chemiosmotic means here the movement of protons inside, that means in the matrix, and coupling means here productions of ATP with the help of this movement. This hypothesis was proposed by Peter Mitchell in 1961. He proposed that this proton motive force is the immediate energy source for the productions of or synthesis of ATP. So he actually emphasized on the proton motive force or proton electrochemical gradient. This is called chemiosmotic coupling. Now during that time virtually all researchers actually rejected his proposals, but despite all the efforts they failed to disprove this hypothesis, and ultimately it has been found that Peter Mitchell was correct, and this proton motive force actually helps in the generation of ATP. So this is called chemiosmotic coupling. So chemiosmotic coupling means this F-class pump can synthesize ATP. That's why this pump is also called ATP synthase. ATP synthase. Now one thing I want to mention is that this is not the only function of the F-class pump. This synthesis of ATP or ATP, we call it forward reaction. Forward reaction. The F-class pump can work in reverse direction also. So they can work in reverse directions. So they can show reverse reactions. In the reverse reactions, what happens? This ATP is break down into ADP and Pi. So you have to understand this. This F-class pump is not only synthesize ATP or they are ATP synthase, but they can also break down ATP and generate proton motive force. So what I am telling about this, proton motive force or proton electrochemical gradients help in the generation of ATP, but sometimes it has been found, specifically in the bacterial plasma membrane, it is frequently observed that bacteria could degrade or destroy, hydrolyze the ATP to make proton electrochemical gradient. Now why it is important? Because for bacteria, movements of or import of sugar and amino acids is depends on the proton gradient. So just in case of mammalians or animal cells, sodium is so important. In my previous videos, sodium glucose importer, sodium amino acids importer, I have mentioned that with the help of sodium movement, glucose and amino acids import inside the cell. The same is true for bacteria. They can degrade the ATP by this F-class pump to generate proton gradients or proton electrochemical gradients. And by the using of this proton electrochemical gradient, they can import nutrients like sugars or amino acids. So this F-class pump not only works in the forward direction, that means synthesis of ATP, or we can call it ATP synthase, it can degrade the ATP to generate proton pumps. So it can work in reverse. the last point I want to mention here, that is the fifth point, is that it works, this F class pump works in reverse of V class pump. I have made a separate video on V class pump, and in that video I have mentioned that V class pump also transport only protons, but there is a difference between the V class pump and F class pump. Generally, this F class pump produce ATP, so we call it ATP synthase, but V class pump always degrade the ATP or hydrolyze the ATP to move protons. I will explain here this fifth point. So I will give you the two examples of, one is F class pump and V class pump. For the F class pump, I will draw the mitochondria. Suppose this is the mitochondria and this is the outer membrane of the mitochondria. The mitochondria contain two membranes that actually surrounds the mitochondria. So this is the outer membrane, and there is an internal membrane also present. Okay, so see, this is the internal membrane or inner membrane. Okay, now in between this outer membrane and inner membrane, this space is called the intermembrane space. And inside this inner membrane, we call it matrix of the mitochondria. So this is example of mitochondria. Okay. As I have mentioned that this F plus pump actually present in the inner membrane. So the FO portion of this F plus pump, the subcomplex is present here, and the F1 is present inside the matrix. Okay, and the proton concentration gradient is actually high outside. So protons are present high outside and protons are present low inside. Okay, and there is a charge separations, like positive ions are present more outside and negative ions are present inside. So protons are actually moving from higher to lower concentrations from intermembrane space to the matrix, and by the movement of this proton, ATP is actually generated from ADP and PI. It makes ATP. Okay. Now I want to mention name of the two scientists. One is Humberto Fernandez Moran, who actually discovered in 1960 that there is a stalk-like structure and spear-like structure. Together we call it F class pump. And after a few years, another scientist, Ephraim Racker of Cornell University, he also actually isolated this F1 the F1 subcomplex of the F class pump, and he observed that if we isolate F1, if we isolate the F1 artificially, then he found that this F1 can break down or hydrolyze ATP. They can break down the ATP, so it can hydrolyze ATP. Okay, but when it is attached with the FO, it generates ATP. So this is the fact he discovered. Now here what is happening, the protons are moving from higher to lower concentration and it generates ATP. But in case of V class pump, this V class pump is present. Suppose this is the lysosome, so this is the lysosomal lumen, and V class pump also has these two subcomplexes. Basically the structure of the V class pump is very similar with the F class pump. It also has a VO segment that is present in this membrane and a V1 segment like the F1 in the cytosol. So this is the V1 and this is the cytosol. Okay, in the cytosol the proton concentration is actually low, and inside of the lysosomal lumen the proton concentration is high. But this V1 actually V class pump, actually what happens, it actually break down ATP into ADP and PI. That's why I have mentioned that this F class pump actually works in reverse of the V class form. In case of V class form, it always hydrolyze ATP into ADP and PI to move this proton from lower to higher concentration. See, here protons moving from higher to lower concentration, showing the downhill movement, and here it is showing the uphill movement from lower to higher concentration. Why? Because to maintain the low pH of this lysosomal pH approximate 4.5 to 5. So to maintain the lysosomal lumen pH very much low, it actually moves protons from lower to higher concentration. That's why it's actually showing the uphill movement, and it requires energy, and this energy is actually provided by the hydrolysis of ATP. So this is the functioning of V class pump and F class pump. I think you have understood. This is the example of mitochondria, and this is the lysosome. So this is the lysosome.